Hey everybody, in this video, I'm going to be going over the 2024 AP Biology FRQs that were just released. And as an AP Biology teacher, I really enjoy doing this every year. So I'm going to go line by line, step by step, and hopefully this video is helpful to you. If you're my student, maybe you can take a look at how you did on the exam. If you are not my student, then I hope and that this video is helpful. It's really important to know that all the answers in this video are not official. These are just my predictions, and the official AP FRQ keys should come out around July. And when they are, I'll put them in the description below. Just a few things before we get started here. And I love doing this every single year. There will be timestamps in the description below. So if you want to skip ahead to any of the FRQs, go ahead and do so. There's also a PDF that's going to be posted below in the description below for you to see what the actual tests look like. And then once again, when these scoring guidelines come out, I'm going to go ahead and post a link to those as well. All right. So hey, just please note this video is created and posted before the release of the actual AP FRQ answer key. But by going through it, the exam, I really hope that this is helpful to you. These answers are my best guesses, so go easy. And yeah, comment below. If there's another answer that you put, maybe they'll take that as well. Because remember, there's multiple answers for every scoring guideline. So yeah, go ahead and comment below, and I'll try my best to respond. And hopefully that's another answer that they took. And then once again, there's no plan for this channel to be monetized at all. My main job and my favorite job is being a teacher. So this is just great practice for me every year to sit down and take the time, just like my students have, to go through the exam and to see what the questions are like, and to help me better help my future students. All right. So, hey, just what's new this year for the AP biology exam? The number of questions is the same as last year, but what's different is the layout. And if you haven't heard already, and as you can see here from their website, the material for the 2 long FRQs are carefully broken down into smaller segments, making it hopefully easier for the student to take the AP exam. Because originally it was a bunch of text and perhaps even a graph. And then below it was a bunch of questions, but now they've chunked it up. So hopefully it's easier to answer from the student's point of view. Something else that's interesting and somewhat new from what I've heard is that there are different versions of the exam. So maybe you took a version of the exam that is not the one that I'm covering right now. The one that I'm covering right now was released by the Collegeboard organization. And it was from what they said, was what was most widely used as the AP exam. So yeah, it says on the website that after 2 days, we can discuss these. And yeah, this is the most commonly used set. I also wanted to show you this as well. I noticed on the website that talked about how, okay, so if there's multiple versions of the AP exam, are they working to keep things equivalent in terms of scoring? And the quick answer is yes. If you want to take a look at how they're doing it, there's a lot more detail on the Collegeboard website. I'll actually link to that below as well. And then finally, one last one. Here's a good question from the Collegeboard website. My students took 2 versions. So apparently that can happen where all of your students sit down to take the exam, and you and maybe the person next to you have different versions. So how is that fair? What's going to happen? Apparently, if that's the case, then the teacher will get 2 different scoring guidelines for the 2 different exams. And from what they said, that and there's a way for them to score it fairly, even though that the questions are different. All right, here we go. Without further ado, let's enjoy. I don't know, for some reason, I really enjoy this process of going through the exams every year. And I really hope that you can get something out of this. All right, so 2024, let's begin. The first 2 FRQs are long, so stay with me here. The first one's especially long. And yeah, so if you're taking the exam, you really got to make sure that you're good with pacing here, because some of these FRQs can take a long time. Let's begin. Crossing over in meiosis 1 is required for homologous chromosomes to properly align during metaphase and segregate during the first cell division. Now, if you haven't taken AP FRQs yet, you'll notice that a lot of these first questions on these FRQs aren't really related to the question. It's just based on what have you learned about biology? Have you learned about crossing over? Have you learned about these different things? Here's the first question. Describe the function of the S phase. The S phase, as we've talked in my class, is part of interphase. And interphase is about roughly 90% of the cell cycle. Once again, the cell cycle is a series of events that must take place in order for the cell to divide, usually from 1 to 2, or in the case of meiosis, from 1 to 4. As you can see, the cell cycle happens before every division, mitosis, or meiosis. And as you can see right here in the S phase. So what happens in the S phase? Here's a response that I would write if I was taking the AP exam. The function of the S phase of interphase is for the a cell's DNA to be duplicated. That's really the answer right there. I always suggest to my students, if you have time, to add a sentence or two after demonstrating to the reader, to the AP reader, that you really understand biology. So it's not just a guess. So here's what I would put. Interphase takes place before both mitosis and meiosis, and the S phase takes place between the G1 and the G2 phase of the cell cycle, explaining that you really understand this topic. All right, so that's A. Let's look at B, but before we look at B, let's take a look at some text that they gave us. Once again, they're chunking it up, and they're giving you text, and then they're giving you a question, etc. Some regions of the chromosome called hotspots, and just so you know, this is a term that I do not discuss with my students as we're learning throughout the year, but it tells you everything you need to know about hotspots. So some places called hotspots display a higher frequency of crossing over than regions. Crossing over is suppressed in chromosomal regions near the centromeres. If you've learned about chromosomes, there's something called the centromere, and that's really what holds together the two sister chromatids. The centromere region of the duplicated chromosome includes a collection of proteins that form a structure called the kinetochore. Scientists hypothesize that one or more of these kinetochore proteins are responsible for suppressing crossing over around the centromere. And as we can see here, suppressing usually means lowering the amount of crossing over events happening around the centromere. All right. To investigate their hypothesis, scientists modified. All right. So once again, we've looked at different examples of crossing over. I just want to pull up some pictures that I've shared widely in my class. As you can see, it's a part of each chromosome that gets switched and that gets given to the other chromosome indefinitely, right? Yeah, so it's going to happen even as the chromosomes separate during the meiosis stages. What you're going to see is a part of each still remain with each other. Here's another picture that I've shown in my class where there can be multiple crossing over events that happen. As you can see, crossing over or double crossing over. But as you can see in the second paragraph, it tells us that in their hypothesis, scientists modified chromosome 8, so just a chromosome, in yeast, such that in each cell, one chromosome from the pair of homologous chromosome 8s contained the gene encoding red fluorescent protein, RFP, while the chromosome from the pair contained the gene encoding green fluorescent protein, GFP. And then it then tells us that cells are expressing RFP emit or give off red light, and GFP emits green light. Now, I didn't know if this was important to note, but I always just like to write down notes as I'm going through this so I don't have to reread it. So on the side, I will write down RFP red light, GFP green light, as you can see in each of the chromosomes, right? And then it tells you that the models are shown in figure 1. So here's what they show you. Here's before crossing over, here's after crossing over. And in my head, because I know I have limited time, I'm quickly looking it over and I'm seeing that, okay, it looks like this chromosome right here has the gene encoding the RFP. It looks like this one has the gene encoding the GFP. But after the crossover, it looks as if one of the genes crossed over and was switched to the other chromosome. Now, as you can see here, each of them usually had one gene, and now this one has one, this one has none, this one has two, this one has one. That's all that's going on in my mind as I'm reading this question. For the second one right here, explain why some haploid cells formed after meiosis in the experiment will only have one fluorescent marker. Well, if you have learned about meiosis, you learned that not every single cell at the end, all 4 of them, have the exact same genes. As you can see, there's genetic variation. There's ways to do that, crossing over, independent assortment, or random fertilization. But as you can see here, that is just what meiosis is. So as I alluded to earlier, in this question, you had to have known that not every single cell is going to have the exact same DNA on the same chromosome. So here's a explanation that I would write if it was my example. Haploid cells at the end of meiosis have gone through 2 cycles of nuclear division, and there's just one of each pair of the original homologous chromosome. So that's the answer right there. I would also explain that in figure B provided, there are 2 sister chromatids that only have one fluorescent marker. So as I said earlier, go back and take a look at that picture if you need to. Then it continues, all right? So we're just getting started on this one FRQ. You've got to move fast on these FRQs. 1 and 2 should take up about half of your time of the 90 minutes, and then 3, 4, 5, 6 should take up about the other half. All right, so we got to keep moving. Here it continues by saying, the scientists then investigated whether attaching individual kinetochore proteins to a specific DNA sequence present in a known crossing over hotspot on chromosome 8 affected the frequency of crossing over at this location. Okay, so what's really important to note, and I would underline this, I wouldn't write it because it would take too long. I would underline that, hey, here's what they're investigating. That if we attach individual kinetochore proteins, will it make a difference? As you can say, will it affect the frequency of crossing over at this location? It then continues by saying, in their first experiment, they examined 3 groups of yeast cells containing the modified chromosome 8. Group 1 contained no kinetochore proteins, as you can see in the graph below. Group 2 contained the kinetochore protein CTF, and it tells you that the CTF, what it does, it's a kinetochore protein, and group 3 contained the kinetochore protein IML attached to the hotspot. For each group, these scientists determined the frequency of crossing over between the RFP and the GFP genes. To determine the frequency, the scientists added the number of cells emitting both. So I took some notes here, and I would just do this in my mind as I'm taking the exam, but group 1, no kinetochore. Group 2 has some kinetochore called CTF. Group 3 has some kinetochore called IML. As you can see at the bottom, it tells you that these are the different kinetochore proteins. So quickly, I'm already analyzing the graph fast, and I'm seeing that, okay, there's none. CTF looks like there's a lower frequency of crossing over, and IML looks like there's a little higher, but look at the error bars. That's already happening in my mind. There's overlap in those error bars. So that's what I'm thinking about in my head. All right, so let's see here. All right, so let's take a look at B. First part. Identify the control group for the scientist's first experiment shown in figure 2. Well, as it already kind of told us, where none is added, that's usually the control group. So here's the response. The control group for the scientist's first experiment shown in figure 2 is group 1 with no kinetochore added. The second one took me a little bit longer than I anticipated. So if it did for you too, I had to reread a few times. That's okay. But once again, in your mind, it's AP exams aren't, in my opinion, too difficult. What makes it difficult is the timing. So you've got to keep moving. So let's read 2, the second part of B. In a follow-up experiment, the scientists created a modified version of CTF in which the DNA binding protein portion has been removed. All right, so I'm thinking of transcription factors. I'm thinking of how it's able to bind to DNA, etc. When compared the frequency of crossing over in yeast cells in the presence and absence of unmodified CTF, okay, so that's unmodified, with that, the yeast cells in the presence or absence of modified CTF not shown. In a follow-up experiment, justify why would the scientists use a modified CTF protein that is unable to bind to DNA as a control group? Okay, so if you don't know what kinetochores are, once again, they're proteins that are in the mitochondrial, in the centromere that is responsible for kind of binding microtubules. So without these kinetochore proteins, as you can see, the binding with these microtubules would not be as efficient or would not be as strong. So here's a quick response. The scientists use a modified CTF protein as a control. Okay, it asks you why. So here's my reasoning to test if it is responsible for suppressing crossing over or not by attaching to a DNA sequence. Well, why? Well, as it said earlier, the modified CTF protein should not affect the rate of crossing over because the DNA binding protein portion has been removed. Right? Because it's modified. It's a modified version of CTF. It doesn't have that. And because it doesn't have that, it should not even do its job. It should not even affect the rate of crossing over. All right. So hopefully that makes sense. The third part, identify the independent variable. My first instinct was looking here and, okay, well, as you can see, the independent variable is what's being changed and here's how we're measuring. But no, you got to read this. Identify the independent variable in the follow-up experiment, not this graph, in the follow-up experiment. In the follow-up experiment, remember what we just said in the point before. It's either modified or unmodified. So the independent variable in the follow-up experiment is the version of the CTF protein, modified or unmodified, in my opinion. Right? There's an answer. Here's the part C of the first FRQ. Based on figure 2, describe the effect of on the frequency of crossing over when CTF is attached to the chromosome 8 hotspot compared to the effect when IML is attached to the hotspot. So my first instinct was, and would be to just say that there's a difference. There's a difference in the effect of the frequency of crossing over when CTF is attached versus IML. As you can see in this graph, we see that there's a difference. But let's elaborate a little more on that to really show the greater that you understand how to interpret graphs. When CTF is attached, the frequency of crossing over based on figure 2, it's about 1% lower. I would use that because it gave you the frequency of crossing over here in comparison to when no kinetic core is attached. When IML is attached, the frequency is about the same as the control group none right there. Okay. In addition, there's an overlap between the error bars, as I said earlier, between none and IML, signifying that the difference is not statistically significant. Whenever there's overlap, once again, even though IML shows that it's a little bit higher, because there's an overlap, we can conclude that it's not that statistically significant. Let's keep moving. We're still on FRQ number 1. It takes a long time, but you got to keep going on the test. Predict the effect on the number of copies of chromosome 8 likely to be present in the resulting donor cells when CTF is attached to the hotspot. So once again, we took a look at the crossing over events here. And as we can see, here's a response. When CTF is attached to the hotspot, the number of copies could increase or it could decrease. So you're like, wait, why? Why is that the case? Well, the second part is provide the reasoning. In the question, I don't know if you caught it earlier, it even told you that crossing over in meiosis 1 is required for homologous chromosomes to properly align during metaphase and segregate during the first cell division. That is a hint to my answer. I would actually rewrite that in the actual answer. Sometimes in the question, it gives you parts of the answer. But I would even elaborate by saying when CTF is attached to the hotspot, CTF will suppress crossing over and could result in non-disjunction. What is non-disjunction? When non-disjunction occurs, chromosome pairs fail to separate, resulting in some daughter cells having more, or like n+1, or some without n. Okay, so that would be my justification to the prediction, because you just don't know which gamete you're going to get. Finally, the last one, explain how the presence of hotspots could increase the likelihood that a population will survive in the presence of selective pressures. Woo! This one right here is asking you, and as you can see earlier, it kind of gave you some information on that. So here, I'm going to take some information from the reading. Because hotspots display a higher frequency of crossing over, that's what it told us in the reading, leading to a higher increase in genetic variation than other regions do, they could increase the likelihood that a population will survive in the presence of selective pressures. Because, remember, selective pressures really affect you when you're not that diverse. But as you can see, because there's genetic diversity, because there's crossing over within the individuals, then it's going to go ahead and increase the likelihood of survival. All right, so hopefully that's helpful. Now let's go ahead and go on to FRQ number 2. All right, here we go. To investigate how increases in environmental temperatures affect the metabolism of certain organisms, researchers incubated liver cells from toads at different temperatures and measured 2 markers of metabolic activity: the rate of oxygen consumption and the rate of ATP synthesis. As I'm going through this, here's just some quick notes that I'm taking mentally, or I'm quickly scribbling down on my paper. The investigation is how increases happen: liver cells, toads, and they told us the 2 measurements that they're going to use. So the first question is, describe the role of water in the hydrolysis of ATP. You should have already known after studying biology that hydrolysis is any single time you're breaking a bond using water. So as you can see, here's a quick answer. Water is used. You got to be very clear. Whenever hydrolysis happens, water is used. Dehydration would be water is removed. As you can see, water is inputted here, is added during the hydrolysis to break the chemical bond between the second and third phosphate of an ATP molecule. The energy is in the bond, but it's in the breaking and the releasing of the energy in the bond that gives the phosphate that opportunity to add energy wherever it needs to go. If you've been studying ATP, that's the ATP cycle. So look up on that if you need to. So from ATP into something called ADP, which is just a carrier. ADP wants to become ATP by adding on a phosphate. All right. So next, it gives us all the data. As you can see, here's some error bars here as well. As you can see, what do I think about? I think of the highest number whenever I'm graphing. Here's just a quick graph that I put together. As you can see, probably shouldn't have used that same symbol for both of them, because I don't know if color is printed on the AP exam, but as you can see, the first bar represents the rate of O2. The second bar represents the rate of ATP synthesis. And it even told you to construct a bar graph to compare. As you can see, not only do we see what the trend is as temperature increases, but we also see the error bars and the role that error bars play. It says, based on the data, provide the temperature in degrees Celsius in which the rate of oxygen consumption is different from the rate of oxygen consumption in 25. So based on the data provided, the temperature 30 degrees has a different oxygen consumption than 25. Well, why did we pick that? Because you can, some of you may be thinking, well, 20 degrees has the same amount of difference. As you can see, it's different than 16.5, right? 12.8 is different than 16.5. But if you do the quick math, using the error bars, at the highest level of 20 degrees, it can reach up to 15. Where do we get 15? 12.8 plus 2.2. At the lowest level of 25, including the error bar, 16.5 minus 2.0, that's 14.5. Do you see that overlap? Because there's overlap, we can conclude that these values are not statistically significant. That being said, we have to conclude that 30 degrees is the answer in that case, right? So the answer is 30 degrees, in my opinion. As you can see, based on the data in table 1, describe the effect of temperature on the rate of ATP synthesis. It will, as we said earlier, based on the data in table 1, as temperature increases, the rate of ATP synthesis in liver cells from toads will also increase. All right, so that's just based on the data table in table 1. Next one, based on the data in table 1, calculate the average amount of oxygen consumed. Now, here's how I got the answer very quickly. It told you in nanomoles for 10 milligrams of mitochondrial protein over 10 minutes. So it tells you 25 degrees Celsius. So immediately my eyes is going here. But I'm taking that number 16.5, and as you can see, here's the units. How do we cancel out the units? Well, by multiplying, right? And the milligrams will cancel, the minutes will cancel, and what you're left with is n moles. As you can see, in this case, it'll be 1,650, I believe is the answer. D, oligomycin is a compound that can block the channel protein function of ATP synthase. All right, so in my head, I'm already thinking about oxidative phosphorylation, the electron transfer chain, H+ ions going from the matrix into the intermembrane space. And when it comes back down, that is how ATP is made. That is the process of cellular respiration. That's how we make all that ATP in the third stage of cellular respiration. So all that's happening in my head. Predict the effects of using oligomycin on the protein gradient across the inner mitochondrial membrane. Some of you immediately may be thinking, oh, it blocks it. Okay, you're right that it blocks H+ ions from going down. But remember, that is only one part of the electron transport chain and oxidative phosphorylation. Oligomycin would actually increase the proton gradient. Well, why would it increase the proton gradient? Well, it's just pretty much testing you if you understand what a proton gradient is. So let me further explain. It says to justify. So here's my justification. Because oligomycin only blocks the ATP synthase, the channel protein function of ATP synthase, and not the other protein complexes. Remember, I believe there's 4 of them, 1 through 4, involved in the electron transfer chain, then the concentration of H+ ions will keep going up to the intermembrane space. So it's going to keep happening. But what's going to happen is now ATP synthase doesn't work, so all the H+ ions are going to stay in the intermembrane space. Hopefully that makes sense. As you can see, the proton gradient definition is the difference in concentration of H+. So as you can see, why is it going to increase the concentration? Because H+ keeps being put up. As you can see here, that's why we answered that it would continuously increase, raising the proton gradient. So take a moment on that, read that if you need to. Upon first glance, you may think, oh, it blocks it, it stops it. You're right, but it only stops one part. And the answers and the questions asking for the proton gradient across the mitochondrial membrane. FRQ number 3. All right, let's keep going. FRQ number 3 says to investigate whether red blood cells of animals lose the ability to take in glucose from the environment as they age. Scientists collected red blood cells from guinea pigs that ranged in age from day 1 to day 7. So as I'm reading this, I'm underlining things. I'm taking some quick notes. The investigation is whether or not RBCs in animals lose the ability to take in glucose. There's guinea pigs, as we said, from day 1 to 7 months. And as you can see, we're going to take a look at the solution of radioactively labeled glucose. As you can see, the amount of radioactive labeled glucose present inside the red blood cells of each group was measured over time. Okay. A asks, describe the difference between passive and active. Well, here's a quick answer. One main difference between passive and active is that passive transport does not require energy because molecules move down something called the concentration gradient from high to low. While we know that active transport pushes things against their concentration gradient, requiring energy because it usually goes from a gradient of low to high, and that energy is in the form of ATP. Okay, so that's the difference between those two. Justify why the scientists use an equal number of red blood cells in each culture dish as a control. Well, we know that you can only test one thing. So I believe that the answer can be scientists using equal number of red blood cells to ensure that only one variable is tested. And that is going to be age, right? That's the one identifiable factor, as you can see, from day one all the way up to 7 months old. The question continues by asking you, glucose transporters are actually required for facilitated diffusion of glucose into red blood cells. That's what it tells you. The scientists claim that the expression of the gene encoding these transporters decreases as guinea pigs age. If the scientists' claim is supported by experimental data, predict the effect of increased age on the amount of radioactively labeled glucose present inside the cells of each group. So here's the prediction, and here's why I think they're being fed. Because the expression of the gene encoding the protein transporters decrease as guinea pigs age, that's what it told you in the question. My prediction is that as age increases, then there would be less amount of radioactively labeled glucose present in the cells. That's the only logical explanation. So slow down on that and pause it and take a look at my answer if you need to. Now, why? So justify it in part C. As guinea pigs begin to age, the expression of gene encoding those transporters will actually decrease, causing there to be less overall glucose transporters available to be used, right? So that gene is going to be decreasing in order to create that glucose transport, all right? So I think that's a great justification. All right, here we go. FRQ number 4. All right, here we go. FRQ number 4 asks, the common wild oat is native to regions of Europe and Asia, but it's an invasive species in Central California, specifically the grasslands. In California, the common wild oat has almost completely replaced some species of native bunchgrass. So you already know that it's an invasive species right there. Researchers found that aphids, a type of small insect that often carries plant viruses, have a much higher reproductive rate in grasslands that include the common wild oat than in grasslands composed of only native bunchgrass species. Additionally, the viruses carried by the aphids appear to affect only the native bunchgrasses and not the common wild oat. Interesting. Native bunchgrasses infected by the virus have much higher death rates than native bunchgrasses that are not affected. All right. So as you can see, here's all that it told us. Here's what's happening. I just underlined or I just wrote down a few details as I do this. A, describe the change in the resilience of an ecosystem when there's a decrease in the number of species. Well, let's take a look at it. Here's a quick answer. I believe that an ecosystem becomes less resilient when there is a decrease in the number of species. More diversity, more resilient, less resilient is because there's a decrease in the number of species. So B, explain how the addition of the common wild oat affects the number of native bunchgrass plants that can be supported by the California grasslands ecosystem. Here's a quick response. Because the common wild oat is an invasive species, so that's what it told us in the question, then it will negatively harm, in my opinion, and lower the central California grassland. And that's what it told us in the question. In addition, the viruses carried by the aphids appear to affect only the native bunchgrasses, causing higher death rates. So we just explained how the addition of wild oat affects the number of bunchgrass plants. Researchers suggest, here's the next question, C, adding ladybugs, okay, and this is a natural predator of aphids, to the California grasslands. Predict the effect of adding ladybugs on the abundance of the native bunchgrass population. So the question told us a lot of what's going to happen. So here's my response. If ladybugs were added to the California grasslands, then the native bunchgrass population would actually increase. Because once again, you're attacking and you're going against the aphids. And in the justification, as I just said, by increasing ladybugs, these natural predators to aphids, aphid populations would decrease and plant viruses would also decrease. Because remember, the aphids, as it said in the question, will carry these viruses, ultimately resulting in what? The protection and abundance of the native bunchgrass population. This one was just reading the question. FRQ number 5. Here we go, almost done. Researchers study mechanisms that enable or prevent speciation. So we learned about pre and post zygotic barriers, things that prevent the zygote from even forming, and then things that happen after the zygote has formed that could complicate things. So A, describe the post-zygotic mechanism that prevents gene flow and thus enables speciation. Well, let's excuse me. Take a look at these 3 post-zygotic barriers that we learned about in our class. So you could have wrote about any one. You could have wrote about hybrid inviability. The zygote, embryo, or offspring produced is unable to survive and dies early. You could have talked about that. You could have talked about hybrid sterility. Maybe the offspring is sterile and it can live, but it cannot breed and it cannot have kids. Or you could talk about hybrid breakdown. Maybe the first generation is okay, but over the next generation, what's going to happen is there's going to be a reduction or prevention of reproduction. So any of those could have answered that question. Let's take a look at the next part. It continues by giving you some more information. New genes, it says, can evolve from non-coding regions of DNA. It is not until certain regulatory elements are present in DNA that a non-coding region becomes a new functional gene that encodes a protein. These regulatory elements include a promoter, a 5' untranslated region, followed by a start. And so it gives you a figure 1 picture right there. Research studied the evolution of the family of antifreeze glycoproteins, AGs, and it tells us down there that what they do is they encode genes in a family of marine fish called cods. When present in fish, here we go. I'm going to take some quick notes here. These glycoproteins reduce the freezing temperature of the fish. The researchers compared genomic sequences in 9 cod species and 1 non-cod species. They recorded the presence or absence of the elements of functional AG genes, as well as AG-like sequences that are similar to functional genes that have undergone that. So it gives you, as you can see here, a phylogenetic tree. It starts over here and then it continues up. But it tells you which ones have the element and which ones do not have the element, absent or present. And then it shows you other genes as well. So here's a question. Based on the data in Figure 2, explain how changes to the genome enabled cods to survive and reproduce after a period of freezing temperatures between 10 and 15 million years ago. So as we said in the question, it tells us what these genes do. And here's a quick answer. Based on the data, cods were able to evolve AG genes, as we saw that earlier, developing an antifreeze glycoprotein to survive through freezing temperatures. These genes reduce the freezing temperature of fish, allowing for better survival. So that's how the changes have helped them survive these freezing temperatures. Using the template in the space provided, place an X on the phylogenetic tree to represent the origin of the function of the AG gene. We'll take a look at it. It starts here. It looks like everything's negative until around here. This species is going to go ahead and evolve into this species, which does not have it. And then this species, which looks to be, begins all the pluses. So as you can see, it's going to happen right there. That's where I would put the X to explain to the reader that you understand how phylogenetic trees work, and that's where the evolution happened. Based on Figure 2, explain how genetic differences among the species in the family of these cods determine the habitats in which they can survive. Here's a quick answer. Differences in genotypes will lead to a greater diversity in phenotypes, resulting in better fitness and survivability for the species of this cod family. So that's how the genetic differences are going to make a difference. Last but not least, FRQ number 6. Let's finish strong. This last one was a little confusing. I'm going to warn you on this first one, but because I was running out of time, even me, on this last FRQ, I just read it quick and I did it quick. Scientists can quantify the rate of translation as ribosomes move along the mRNA. We know in protein synthesis that ribosomes are going to go and create proteins through transcription and translation, but here it talks about the rate of translation amongst that from one codon to the next. Using a procedure called ribosome profiling, these scientists measured how long a ribosome remains stationary at each codon. They determined the average translation rate across the codons is about 5.2 amino acids per second, and that the average translation rate varies widely. These variations in translation rates are thought to facilitate correct folding. As you can see, the rate was measured in 100 different mRNAs, and then it gives us this data. Quickly, when I'm reading this data, I see GAC on top, AUU, and UGG. There's a difference in all 3 of these codons in the translation rate. As you can see, this is MS per codon. So as you can see, this one's going to take this long. This one's going to take this long. This one's going to take a lot longer. Those are the trends that are happening in my head. Here's the first question. Using the data in Figure 1, Graph A, identify the rate in MS per codon that was recorded the greatest number of times for GAC. Here's a GAC, and as you can see, that's what I'm looking at right there. The rate that was recorded the greatest number of times in the that should be GA. GAC, I'm sorry, that's GAC coding, was 150 ms per codon, millisecond per codon. All right, so that's the highest. So that one's pretty straightforward. B, using the data in figure 1, graphs B and C, describe the variation translation rate of the AUU compared to the UGG codon. Here's my quick answer. UGG had a higher average rate, as you can see in the graph right there, as well as a higher maximum translation rate. That one goes up to 950, whereas AUUs go up to 450. So that would be my quick answer to describe the variation in translation rates between AUU and the UGG. I believe this is the last one, right? Oh, two more. Scientists hypothesize that tRNA molecules that bind to the UGG codons are available in lower abundance than are tRNAs that bind to AUU. Support the scientist's hypothesis using the data, figure 1. The scientist's hypothesis is proven that the data in figure 1, looking at the graphs B and C, as you can see right there, the average translation rate, ms per codon, for UGG is higher. Take a look at it. The average rate is higher in that graph than it is AUU. This translation rate can be explained by the lower abundance of available tRNAs. That would be my best guess. I don't know if that's right, but that would be my best guess. Last but not least, amino acids can be encoded by multiple codons and many things. Explain why the use of one codon over another for the same amino acid might result in increased levels of protein production. This one tripped me up a bit. Here's my quick answer. The use of one codon over another for the same amino acid might result in increased levels of protein production. Why? Because codons, as shown in the data provided, are translated at different rates. So the graphs tell us that they're translated at different rates. Codons that are more prevalent, common, will have more tRNAs available, making the translation process quicker. And if you make it quicker, think about it, you're going to increase the levels of protein production. So real quick, I just want to say thank you. I really hope this was helpful. I wish you the best on your AP exam. If you already took it and you didn't put my answers, it does not mean that you were wrong. There could be multiple answers. And remember, it's just one test of many, many tests that you're going to have. Everyone, I really believe, has a great future ahead. The key thing about learning is just enjoy the learning. Keep practicing, keep growing, keep going. So reflect, grow, and continue on your journey. And thank you so much for watching. Once again, I just hope that this video is helpful. I truly enjoyed doing this, and I hope to continue doing this for many years to come. Thanks so much. See you next time.